Oh my gosh.
Hello. Yay! I love that.
Hi, Erin.
How are you?
Doing okay. How are you? Feeling better?
Yeah.
How was, Maddie told me you had a comedy show on Saturday. How was that?
Yes, it
Fun.
was good. Mike actually got picked on, so I think Mike's gonna
On TikTok? Why do you think?
Because like
Hey everyone. Hi. Big crowd here so far.
the comedy show that we went to, there's like crowd work, so it's not like a normal. Like he doesn't like say a bit.
It's not like a set.
Yeah, he just goes to the crowd and like takes up.
I don't know why it's like
Oh, you're back there.
So he like we sat in the front on the corner of like an aisle, like went down the aisle and it was like a bit about not having a dad. So he asked Mike, is dad?
Whoa.
So yeah.
Okay, good.
Where's she?
I didn't finish it. I just needed the solid color from the background.
Is it weird if I hold it up?
No. Is that comfortable for 30 minutes?
No, because I'll go like
No.
so I'll pin it like here.
Oh,
Is that good?
you're supposed to something. Wait, repeat that.
Like this? No.
Oh, wait.
Where am I supposed to put it?
On your
Like
collar.
up, but then it's gonna be against my neck.
Can you angle the little microphone out?
No. It's not better?
That's better.
Oh. Okay.
You're pretty loud.
I'll keep it here. Well, I'm gonna unclip it for now because I don't want people to. So Aaron has to talk to me. Unless you want to.
I can shout.
You can shout.
I did for my video.
Oh. Your proposal?
I didn't know that.
I now have a lifetime supply of those microscope slides.
Oh, really?
Like a giant box, because that's all the Fisher would sell.
The charge ones?
Charge. 1,400. For $600.
1,400.
Oh, really?
Yeah.
Because you go through a lot.
You go through a lot. For you, though, it's probably lifetime supply. Yeah.
Well, I think for girls managed to get figure out how to use that.
3 hours?
Yeah.
Certain slide and test out that sequence protocol.
Yeah. Right.
Is that what Keith worked on?
Yeah, but you know, like couples who are in fish. It's just like one player.
I'm not in her.
But it's, I mean, it's gotta work. That's I feel like most of the
I was like, dude, that's too far. He's like, you're right, you could drive.
we'll see. So like my psychology.
I'm on the car.
I may send some equivalent from talk to you some more.
Yeah.
Yeah.
I think it's hard for the
Yeah.
You know, you want to make them
strings.
I don't know.
Do you have any?
Yeah, I think it's different.
That was really fun.
Yeah.
while there's persevering,
Yeah, that's
I was thinking of something like
works on their fixed missions, exactly true.
right. No, I
This is a bad idea.
Kind of works, but it really doesn't like fixed missions. So there's like a lot of optimization that's going on to get it so that the antibody work. You know, the cells are fixed, but your antibody works in the same time. So it's definitely not trivial.
Okay.
So
You mentioned these two chips.
yeah, I don't know. I don't know. Is there a reason you think this will be better?
I thought it would be higher resolution in chips.
I don't. If you can get it to work, like we should be prepared. Not like once optimization. Okay, like if we just want the data now.
Should we start?
Good.
I'm gonna introduce Emily. So it's my pleasure to introduce my friend Emily. She is a third-year PhD candidate in Dr. Philip Hughes' lab, where her research focuses on the influence of metabolism on epigenetic programs in memory. Prior to joining BU, she worked at Mass General as a research technician, and this past year she was awarded a F31 fellowship. Today she will be presenting her work titled Metabolic Regulation of Histone Methylation in Memory Encoding.
Thank you, Erin. And thank everyone for being here. So like Erin said, I'm in Philip Meade's lab, where we work kind of on this metabolism epigenetic axis. So in the Meade's lab, we work in the brain. So when we think about how a memory is encoded, upon a neuronal stimulus, we get activation of a subset of neurons termed an ensemble, and through repeated stimulus in recall events, we actually get re-engagement of those same neuronal populations, eventually resulting in an encoded memory, and this is referred to as a memory engram. These neurons that get activated see massive upregulation of immediate early genes, or IEGs. IEGs are rapidly transcribed upon neuronal stimulation, and then they go on to regulate secondary response gene programs as well as modulate synaptic plasticity. So when we think about disorders of learning and memory, there's kind of two avenues that my talk will focus on today. The first one being this formation of what I'm calling a weak ensemble, which is things like dementia and neurodegeneration, where you get sort of cognitive impairment. The second one being this enhanced ensemble, which is actually strengthening of that memory engram to kind of a pathological point. And this encompasses things like substance use and neuropsychiatric disorders. Specifically today, I'm going to be talking about cocaine use disorder. So the way that cocaine works is it enhances dopamine signaling in the brain's reward circuit. So first I'm just pointing out these two key brain regions, the nucleus accumbens and the hippocampus, that I'll be focusing on. But for right now, we want to talk about the NAC. So the way that the dopamine signaling happens is you get dopaminergic inputs from the ventral tegmental area into the nucleus accumbens. And the NAC is comprised of two kinds of medium spiny neurons, just differing in the type of dopamine receptor that they express. So D1 is type 1 dopamine receptors and D2 is type 2. And it's widely accepted that activation of D1s actually enhances motivation behavior, while activation of D2 suppresses that motivation behavior. And what we know about cocaine is actually cocaine leads to really enhanced activity of D1 medium spiny neurons specifically. And this is just a simplified graph of calcium imaging studies. What's also notable is that the pool of D1 medium spiny neurons that are activated is actually very small. So this is just a UMAP from single nuclear RNA-seq data showing all D1s, and then what's highlighted here is actually what we're calling the D1 IEG cluster, because they upregulate all the classical immediate early genes that we think of when we think of neuronal activation such as ARC, FOSB, and NR4A1. And these gene programs are facilitated by changes in chromatin packing. So this is just a simplified schematic of the way that our DNA is packaged inside the nucleus. So we have our DNA wrapped around histone optimers, and this is a good example of euchromatin, so things are really open and accessible. On the flip side though, we have heterochromatin, where things are a lot more condensed and packed in, and this has a big effect on gene expression because genes need to be accessible to become transcribed. And the way that these changes occur is actually through histone modification. So histone modifications are chemical tags that occur on residues of histones, and these include things like acetylation, methylation, and phosphorylation. And these are put on by classes of enzymes called writers. There are other enzymes that can actually bind to and identify certain histone modifications and then recruit certain cofactors such as chromatin remodeling complexes, etc. And then there are also enzymes that can take off these modifications called erasers. So these modifications can be inducible, like we see with cocaine exposure, but actually the effects they have on chromatin structure can actually be quite long-lasting. So here again, I'm showing just this activation of neuronal ensemble with an initial cocaine stimulus. But actually what we see in a withdrawal period after there's no cocaine being given is this sort of primed phenotype where you actually still get accessible chromatin without the genes actively being transcribed. So that way, what we think happens the next time there is a cocaine event, you actually can get really rapid high inductions of these gene programs, and maybe even stronger than the ones before. So this whole priming of the chromatin that I'm describing is regulated by epigenetic mechanisms. So my projects focus on histone methylation, and histone methylation is interesting because it can be both like dynamically induced by certain stimuli, and it can be very long-lasting depending on the modification you're looking at. Importantly, it can also be activating or repressive. So this can occur on lysine residues as well as arginines, and methylation can either be mono-di or trimethylation. So when we think about activating methylation, the pretty classical one is K4-trimethyl. So this is trimethylation of lysine 4 on histone 3, and this occurs mostly at promoter regions. This is put on by classes of, or a subset of writer enzymes called histone methyltransferases. And this whole process is actually very closely tied to metabolism, as SAM, or S-adenosylmethionine, serves as the universal methyl donor for these reactions. SAM is synthesized by MAT2A, or methionine to adenosyltransferase 2A, and this kind of role of metabolic enzymes in regulation of epigenetic landscapes is a really novel and exciting sort of regulatory mechanism that's just now starting to be picked up a little bit. But actually the role of MAT2A that it has on methylation is actually pretty underexplored, especially in the brain. But what we do know actually is that in those same D1 medium spiny neurons that see upregulation of those immediate early genes, we also see massive upregulation of MAT2A in these cells. So that begs the question, is there a role for MAT2A in neuronal activation? So the first thing we wanted to do is see if MAT2A localizes to the nucleus and neurons. So we started off with a differentiated N2A model, which is just mouse neuroblastoma cells that we differentiated into neurons. And by some IF, we can see that MAT2A is expressed in the nucleus. So we have our green MAT2A over here. And we can see co-localization with our DAPI-SAM, which stains our nuclei. We also wanted to know if neuronal activation impacts MAT2A expression, especially since we see that upregulation in vivo with cocaine. So what we did is stimulate our cells with chemical long-term potentiation. And by RNA-seq, we actually can see massive upregulation of MAT2A alongside the canonical immediate early genes we'd expect, like JUN, FOS, and NR4A2. And then, does MAT2A regulate activity-induced genes? To answer this, we again stimulated our cells with that chemical long-term potentiation. But we actually also gave them AG270, which is a small molecule compound that inhibits MAT2A catalytic activity. And what we found here is that activity-induced gene expression is actually inhibited with the addition of this AG270 inhibitor. So up here you can just see the genes that are upregulated with stimulation, but then when you treat with AG270, you see downregulation of those programs. So this leads us to a key mechanistic question, which is, does MAT2A localize to chromatin to provide SAM for histone methylation? So the first question, does MAT2A bind to chromatin in neurons, is obviously very important. So what we did is simulated our cells and then performed a simple subcellular fractionation experiment. So we just used a fractionation kit and then ran Western blot for MAT2A and quantified that. And what we see is that MAT2A is found in both the chromatin and the soluble nuclear fraction. And actually, if you stimulate with CLTP, you can increase the ratio in the chromatin fraction of that nuclear MAT2A. Now, where does MAT2A bind? To answer this, we used cut and run against MAT2A. And we actually did see that MAT2A is binding to promoter regions of activity-induced genes, ARC and FOS. And actually, this binding overlapped with known H3K4 trimethyl regulated loci, which is that activating mark I talked about before. We also wanted to know if MAT2A was interacting with any chromatin modifying enzymes. So we did this technique called RIME. RIME, you cross-link all your proteins in your DNA together, and then you use antibody conjugated beads to fish out your protein of interest. In this case, it's MAT2A. And then you actually digest off all the peptides that you fish out with MAT2A and run mass spec to identify its protein interacting partners. And this assay actually worked really well. Our top hit was MAT2A, as expected. The second hit was MAT2B, which is the regulatory subunit. And we actually also got a whole handful of methyltransferases, I'm just pointing out two, SET-D1A and SMIDE5. And if we ran molecular complex detection on these hits, you see things enriched like chromatin looping, regulation of chromosome organization, and stuff like that. So we can conclude that MAT2A is interacting with histone methyltransferases at chromatin. So since we saw this MAT2A chromatin binding in our tissue culture model, we wanted to see if this held true in vivo. So we moved into the mouse nucleus accumbens, and I performed MAT2A ChIP-seq. And what we found is that, yes, it is binding to chromatin in the nucleus accumbens, and it is also binding at gene regulatory regions, mostly promoter-driven. We also see binding at activity-induced genes such as EGR1, so similar to what we saw in the tissue culture model. And actually what we can see here when we look at saline versus cocaine is that when you activate that region with cocaine, you actually see increased binding, which correlates nicely with our fractionation data. And this is also seen by just the total amount of peaks that occur in saline-treated animals versus cocaine-treated animals. So we see more peaks, concluding that cocaine leads to increased MAT2A recruitment. So if we do Go analysis on the MAT2A-bound genes, we find that it's enriched for things like epigenetic mechanisms as well as genome maintenance cores. These are shared between the two treatments. But actually, if we look at the genes that are differentially bound between saline and cocaine-treated animals, we see a lot of shared things, especially on the epigenetic side. But we also see amplification of this genome maintenance core, which we think may be suggestive that MAT2A is facilitating some sort of response to the stress the cell undergoes from the cocaine stimulation. So those genes are being bound by MAT2A, but if they're actually regulated by cocaine on the transcriptional level is a different story. So we are actually able to compare that data to this other data set that has been recently described. And these mice, so they were done, this data was done in these transgenic mice, which allow for fluorescent labeling of D1 and D2 nuclei, and then you can do cell sorting. So this is from a single nuclei RNA-seq data set where mice were treated with either chronic cocaine or saline. put through a 30-day withdrawal period, and then either injected with an acute dose of cocaine or saline as a control. So this allows us to kind of parse out the transcriptional responses from D1s versus D2s with single nucleotide resolution. So actually, when we overlay that data with the peaks that we know are bound by MAT2A with cocaine, what we see is actually, so this is all from MAT2A bound genes, we found that in D1s, these genes were transcriptionally upregulated with cocaine, but this was actually not observed in D2s, allowing us to conclude that MAT2A bound genes are induced by cocaine in D1 medium spiny neurons. So here I'm just showing that yellow is upregulation, and you can see here with acute cocaine that kind of matches the challenge cocaine, but you don't see that effect in the D2s. So we also wanted to know if MAT2A is supporting this cocaine-induced gene activation. So what we did is knock down MAT2A in the mouse nucleus accumbens, just using stereotactic surgery and viral vectors. Waited 2 weeks for viral incubation, and then injected an acute dose of cocaine or saline, and then did bulk RNA-seq. And what we found actually is that MAT2A knockdown blocks the induction of these cocaine-responsive gene programs. So up top here in wild-type cocaine, you just see the genes that are differentially expressed with cocaine treatment, but actually when you knock down MAT2A, you get dysregulation of these programs. We also wanted to test if oral SAM supplementation can rescue this knockdown effect. So SAM, as a reminder, is that metabolite that MAT2A produces. So what we did is we knocked down MAT2A, but before cocaine administration, we actually also supplemented the mice with an oral lavage of SAM, and then performed RNA sequencing. And actually what we found is that, so we see those same differentially expressed genes with cocaine, we see that dysregulation by MAT2A knockdown, but then when we supplement with SAM, we see a counteraction of that knockdown effect. So that SAM supplementation is rescuing the effect of the MAT2A knockdown. So that kind of brings me to the next steps for this project. So we want to correlate some of this MAT2A binding with chromatin accessibility, so we have plans to do some ATAC-seq there. We'd also like to get some functional readouts, so hopefully we can relate this back to just neuronal activity in general, so we'd like to get some EFIS done. It also would be great to correlate our findings with behavioral readouts, so a big part of cocaine is that seeking behavior, of course. So there's a couple different behaviors we can run. One is conditioned place preference, where you condition a mouse to know that they will receive cocaine on one side of a two-sided box, and then you test which side of the box they want to spend more time on. The other one is self-administration, which is a volitional model of cocaine, in which the mice have exposure to two levers. One is active and one is inactive, and when they learn that when they hit the active lever, they actually get a dose of cocaine. So it'd be interesting to see the effects of our MAT2A knockdown or a MAT2A inhibitor, and if they have any effects on the cocaine-seeking behavior. Okay, so that brings me to the end of the addiction side of things. So now I'm going to move on to the sort of weak ensemble that I talked about before, that comprises things like neurodegeneration, dementia. And this is what I like to think of as like a weakened ensemble, because you get that cognitive impairment. And because we know at least that MAT2A is regulating things transcriptionally in the brain, I wanted to know if MAT2A dysregulation can affect memory in any way. So what I did is I knocked down MAT2A in the mouse hippocampus. So we moved into the hippocampus, which is a brain region really important for learning and memory. There's a lot of plasticity that occurs there. And I put the mice through a few different behavioral assays that test memory. So I did confirm efficient knockdown of MAT2A by Western blot when compared to scrambled controls. So we see this quantification of MAT2A here, as well as the expression of our GFP reporter. We also saw a strong trend in reduction of H3K4 dimethylation, which is K4. It's kind of activating, it's at promoters and enhancer regions, but we can say we're hoping to add more animals to this, but we're hopeful that MAT2A knockdown results in these methylation deficits. So the behavior, so we tested them for recognition memory. So this is just testing if the mice can recognize that there is a new object that they haven't seen before and just kind of scoring how long they interact with that new object. And we did see a significant deficit there with our match-weight knockdown animals compared to controls. We also tested them in contextual fear conditioning. So you put the mice in a box, they get a foot shock, and then the next day you put them back in the box and see if they can remember that, oh, I was in this place, I'm scared, and then you kind of measure freezing behavior. So what we saw there is, again, a significant deficit in the contextual fear conditioning with our knockdown mice. We also tested them for location memory, so just seeing if they can remember where they saw a certain object in space. And we saw a trend there between our control and match-weight knockdown animals as well. So we can conclude that match-weight knockdown results in memory deficits. So speaking of memory deficits, I think the most classical example of this is Alzheimer's disease. In addition to the protein aggregation phenotype that we see in Alzheimer's, we actually also see severe dysregulation of one-carbon metabolism, of which the methionine cycle is intertwined. So notably, we see that SAM, which is that metabolite, is down in Alzheimer's human brain, pretty much across the board in every relevant brain region. We also know that AD patients and models exhibit this DNA hypomethylation phenotype, where actually, if you supplement with chronic SAM, you can rescue this hypomethylation. But actually, how this process regulates histone methylation and subsequent gene programs is pretty underexplored, so that's what my project is focusing on. So in order to answer this, we are using the APP-SAW knock-in mouse model. So this is a knock-in model of Alzheimer's disease. It has a humanized APP sequence as well as 3 familial AG mutations. These mice exhibit robust amyloid beta pathology, and they also exhibit the metabolic deficits that we see in Alzheimer's patients. So this is just metabolomics data from microglia of this mouse model actually, but you can see a difference in the metabolite profiles of A beta negative versus A beta positive microglia. So this is a pretty newer model, so I just wanted to characterize it a little bit to start. So it's important, obviously, that these mice can recapitulate the cognitive deficits that we see in Alzheimer's. So I put them through some memory behavior and actually found that these mice do have memory deficits at 6 months, specifically in the contextual fear conditioning that I talked about before. So this is really exciting because I now have a phenotype to go off of. I also wanted to know if these mice have perturbed histone methylation, so I harvested hippocampal tissue from these mice at 6 months of age and actually found a strong trend in reduction of H3K9 trimethylation compared to age-matched controls. K9 trimethylation is a canonical repressive mark, and it also can be induced with neuronal stimulation. And it is known that both activating and repressive methylation is super important for memory consolidation as a whole. Finally, we know that SAM supplementation does have some therapeutic benefit to it. So I wanted to test if MAT2A overexpression in specifically neurons of the dorsal hippocampus could kind of mirror this effect that we see of SAM supplementation. So I overexpressed MAT2A in neuronal nuclei of the hippocampus in these mice at 4 months, which should be before severe pathology onset, and then analyze them through behavior and molecular analysis at 6 months of age. So I did confirm good overexpression of MAT2A in these animals. So down here we have our endogenous MAT2A band with our controls and then our APP-SAW mice. But the rightmost lane here is our overexpression mice. So we see that nice strong overexpression band, and it's just a little bit heavier than the endogenous just because of that addition of the GFP and B5 tags. I do also see enhancement of freezing behavior in contextual fear conditioning in these mice with the MAT2A overexpression compared to non-surgerized APP-SAW controls. So I'm actively adding more animals to these analyses, but I'm hopeful that this upregulation can enhance hippocampal memory in these mice. So to conclude, we know that MAT2A is binding to chromatin in the mouse brain, and it's binding actually at plasticity-related genes. We know that dysregulation of MAT2A, especially in the hippocampus, can result in cognitive deficits. And we also know that upregulation of MAT2A in the APP-SAW Alzheimer's mouse model can enhance memory. So for some future directions, I'm currently performing CHIP in the APP-SAW hippocampus to map that MAT2A genome binding. Henry, yes, do you have a question?
Yeah, but finish.
Okay. I'm also going to include some histone methylation marks in this data as well, so that maybe I can correlate MAT2A binding more or less with one of those methylation marks. I also want to know, how does MAT2A regulate gene programs near amyloid beta aggregates? So I'll be using spatial transcriptomics to manipulate MAT2A levels close to and far from amyloid beta and see how they can alter a neuron's response to stimuli. I would also like to know if SAM supplementation can rescue molecular deficits as well. Yes, Henry?
Sorry, I don't know anything about the brain. In your EE mouse model, how did you accomplish MAT2A overexpression? And is that targeted to just neurons, or is that hit a whole?
Yeah, so we did stereotactic surgery where we have an AAV vector where we can overexpress MAT2A, and then it uses a CAMK2 promoter, so we can drive that overexpression in neurons specifically. So we're doing that at 4 months, and then you just have to wait a couple weeks for the expression to ramp up, and then you can get that nice overexpression band here.
And for your Western, is that just like purifying neurons, or is it like?
No, so this is just like whole cell lysate from a hippocampal punch. I actually was surprised that we saw this much, because I was like, maybe we won't see really strong overexpression here. But I think it's important to note that the hippocampus is like really enriched for neurons. So we saw really good overexpression there. Yeah. Thank you. Thank you to the rest of the MEADS lab, of course, for your contributions to this work. I'd especially like to shout out Hiroshi, who did a lot of the in vitro stuff and has been really, really helpful with all the cocaine work. Thank you to the PPP department, as well as the GSI, the program, and our funding sources. And I'll take any questions. Yes.
Emily, that was great. And great talk, and I have 3 questions.
Okay.
First question is, how did you guys do the RIME assay? Was it in-house, or did you go with a company?
So we did the bench part of the RIME ourselves. Actually, Hiroshi was the one who completed that. But then we did send the digested peptides out. We worked with there's a mass spec core actually at Yale that we worked with to do the mass spec analysis. Yeah.
Okay. And then I have a question about MAT2A. When you knock it down, can you measure SAM levels going down in that mouse where those cells are knocked down? And then the second question is, like, when MAT2A, when you do the MAT2A chip, is MAT2A directly binding to histones, or is it binding through those other factors in your RIME data?
Yeah, good questions. We definitely could. So I think it would be really good if we had, if we could validate the knockdown also resulted in lower SAM levels. We definitely could do that with maybe a metabolomics approach, or there are also like SAM ELISA kits that we might be able to utilize for that. So we might do that in the future, because that would be really good to correlate. And then the second question, so I don't think that we think that MAT2A is directly like binding, especially to DNA. I think that it probably depends on whatever it's complexed with to be in the vicinity of chromatin. So actually to do the chip, as well as the cut and run, we double cross-linked everything together. So we used DSG to cross-link our proteins together, and then formaldehyde to cross-link our proteins to the DNA. So I think we're depending on these protein-protein interactions to actually get it where MAT2A is in space at chromatin.
Oh, okay. So then what defines its specificity? is it has to be those other factors then, the CETDP1?
Probably yes. Yeah, I think it would be, well, I think it's important to note also that we got methyltransferases in that RHYME hit list, but we didn't get like everything. So I think there is specificity to it. We didn't get all methyltransferases. We didn't get like acetylation enzymes or anything like that too.
WDR5.
I don't believe so. Yeah. But I think that our hypothesis is that there are maybe some methyltransferases that actually depend on that localized SAM production more than others. So I think actually the RHYME is hopefully a really good indicator of which sites those are. And then if we can ship or do cut and run for the modifications that those regulate, maybe we'll see overlap of MAT2A binding there with whatever the chromatin modifying enzyme is.
Do you think MAT2A, is it important for it to be on chromatin? It still will have its function if it's not.
Yeah.
Right? So it's, you have the functional results, it's good. And, so
right.
What is the significance of that for you?
Yeah, so MAT2A, we had to test if it was in the nucleus of these neurons. So it certainly has a whole slew of other functions. I mean, it just makes SAM, but SAM is used for a plethora of things. It's used for neurotransmitter synthesis, phospholipid synthesis, all this stuff. So I think that is why we chose, at least in our overexpression experiments, to make sure that that was nuclear overexpression specifically. I think also further down the line, once we have more of a mechanism, we could certainly do some rescue experiments as well to validate that like this is actually the causal mechanism. I think it also would be interesting if we could, inhibit MAT2A from going to the nucleus if that's what it's doing. So.
Yeah, but it doesn't have to be bound to chromatin to those specific sites, right?
Right.
So you don't have to push yourself. You are not dependent on that.
Yeah.
It will be your story will be still fine.
Yeah.
If you just rescue it.
I do think that depending on, so the different methyltransferases actually also do have different affinities for SAM. So part of our hypothesis is actually that different methyltransferases, like I just mentioned, will just be more reliant on local SAM, so they can just grab it and methylate their substrate right away. So maybe that mechanism will be important for at least the transcriptional regulation, but we'll see. Yeah.
I have a question, but I want to first offer my questions to our test student. Should I go?
Yeah.
I have a super weird question.
Okay.
So folate deficiency, folate pathway, and methyltransferase pathway have some shared features.
Mm-hmm.
And folate deficiency leads to neuromuscular defects in development.
Okay.
And so in response, there have been a number of public health initiatives to fully supplement a bunch of things in westernized foods.
Okay.
So I'm curious if folate is another compound you might test, and if that might be a public health way of increasing numbers of various forms of infection.
Yeah. Yeah, that's an excellent question. So actually, if I go back to this cycle, so 1-carbon metabolism, super big and complex. There certainly is a lot of work actually already being done with like B vitamin supplementation, folate supplementation, choline, what have you. That is actually in the dementia space already. But what is also interesting is that SAM seems to be down in the human brain, so is SA, which is just the byproduct after the methylation reaction. But along this pathway, you at some point get some sort of like clog almost in the system. So we were wondering if the upregulation of MAT2A activity can actually just like flush everything through the rest of the system and restore methylation profiles. But it's certainly worth exploring if supplementation of different metabolites in the cycle could have similar effects. I think so far, what's been done in this space is more so just cognitive and pathological readouts and not a lot of molecular characterization with when you manipulate those systems.
I wonder too, since neural tube defects are fairly extreme, I wonder if there's a less extreme kind of phenotype that doesn't come up until later in life that might be related.
Right. Yeah, that's certainly a good idea. Yeah. Thank you, everyone. Thank you. It is, yeah.